So as we were discussing, we can say that it is a sauerkraut itself. So we can say that it is actually the finally cut. We can say that finally cut raw cabbage, okay, which is actually been fermented. So we have to remember that this is actually been fermented, okay, by multiple types of that of the lactic acid kind of that of the bacterium itself, okay. Now, what is the major like advantage for that of this thing? The sauerkraut, it is actually what? It is actually, I would say that it is mainly utilized for that of shelf life extension itself, okay, along with in order to produce that of distinctive flavor, the aroma, and all these things, they can be produced over there itself in that case, okay. Moreover, we have to remember also that it is actually the national list of Germany, just as I said you, that these kind of the products, they have been mainly produced in the foreign countries itself, and they are the national dish of the Germany itself, which has been utilized. Now, if I'm saying about the microbial flora, so what is a microbial flora? Okay, the first phase of fermentation that is being utilized in the sauerkraut itself, it is actually done in that of three different phases. The production of the sauerkraut can be done by the three different phases. In the first phase, there is the involvement of that of certain typical kind of that of the anaerobic bacterium. Okay, we can take the example as that of the Klebsiella as well as the Enterobacter itself. And in that of the second phase itself, we can have Leuconostoc mesenteroides, okay, as well as that of the Leuconostoc sp itself. And in that of the third phase, we can have Lactobacillus brevis as well as Lactobacillus plantella. These things that we can have over here. So all these three phases, okay, the first phase, the second phase, and that of the third phase, all these are what? All these are utilized in that of what? All these are utilized in order to produce that of the sauerkraut itself. And when the sauerkraut are being produced, they are being utilized in that of like in order to extend that of the cabbage itself in a pickling kind of that of the format itself that will be getting formed. Okay. Now the next component, a very favorite food for all of us, okay, mainly having the South Indian origin, and now it got spreaded in throughout our country, that is the idli itself. And if you are saying the preparation mode for that of the idli is generally prepared by that of, we can say that by fermentation of that of a mixture of that of gram and that of the rice itself, the black gram as well as that of the rice batter, okay, by that it can be prepared. It is mainly prepared itself, and the proportion of that of the rice to that of the black gram, it is always 1:4 or to that of the 4:1 itself. And we can say that the black gram, what is the process in that one? The black gram as well as that of the rice, they are then soaked within that of like in separately within that of the water, okay, for approximately 3 to 10 hours, okay, prior to that of like 3 to 10 hours itself. Now when it got soaked itself, after that what we are doing, after that we are taking out this gram, okay, the black gram, okay, and there are the rice, and then we are making it the paste itself, okay. Now when the paste has been made, now these pastes have separately been made, and after that they are been mixed up with each other itself, and there been the application of the salt as well as that of the like enough quantity of water, and it is left aside. When it is left aside for certain period of the time itself, like 18 to 30 hours approximately, automatically the microbial flora, the natural microbial flora will get developed over there, and it will be making it fermented, and it will be getting its typical flavors itself in that case, okay. And this is a rich source of that of the carbohydrate as well as that of the proteinous kind of content, along with that of many different time of the many different types of that of the like vitamins also, it will be getting produced over there itself, okay. So what are the different microflora that are associated with the fermentation? Bacteria as well as yeast, that means fungus as well as microbacteria, both of them are utilized over here. And in terms of bacteria, we can have Leuconostoc mesenteroides, lactobacillus different species itself. And in yeast we can have Saccharomyces as well as Trichosporon, Debanoromyces, and all these part itself, okay. So ultimately what is happening, we are taking the dal as well as we are taking the rice, where separately we are making them soak within the water, and after that we are simply making the paste, okay. And when we are making the separate paste itself, we are making both the paste gets mixed up with each other. When both the pastes are getting mixed up with each other, they are been like salt is applied as well as some amount of water is sprinkled over that one, and they have been allowed to ferment for certain period of time itself in that case, okay. So the batter, it will be then allowed to ferment for that of 18 to 30 hours approximately by the mean of that of the natural microflora itself, okay. So if I'm saying about that of the dosa, Okay, in terms of the dosa itself, we can say like prepared by steaming as well as fermented black gram, okay, and rice itself, the batter, okay, both of them, same thing, it will be getting formed also. Now, what is the nutritional value? As I said, both the dosa and that of the idli, both of them, they are consisting of that of, they are produced actually with that of the very lesser amount of that of the oil itself, or very less amount of that of the oil itself, okay, and they are raising up the vitamin B as well as the vitamin C content of that of the food itself. Okay, as I said you, it is consisting of sugars, okay, but no additional sugars. Whatever the sugars are remaining present, all they are the natural sources of that of the sugar. It is not an additional sugar which is remaining present over there. Okay. Now, if I'm saying about the other microflora, okay, which are associated with that of the fermentation itself, same thing, bacteria is the Saccharomyces, the Leuconostoc, Lactobacillus, different form of that of the Lactobacillus itself. But in terms of the dosa batter, the typical flavor has been brought about because of that of the Bacillus amyloliquefaciens itself, a bacterium, okay, because of which the typical flavor, the typical aroma that has been brought about. Okay. Now, in terms of that of the yeast itself, we can have the Saccharomyces cerevisiae, Libromyces hansenii, and Trichosporon baegelii. These are the majorly utilized the dosa batter like part itself. Okay. So these are the major kind of the fermented foods which are in discussion. Now we need to see that how the what are the different roles of that of the biotechnology in that of the agriculture as well as that of what, as well as that of, I would say, in like food production itself and everything. And in that case, first of all, we'll be taking up the productivity of that of the plant. So if we want to understand like how biotechnology is involved in increasing the productivity of that of the plant, biotechnology is first of all, it helps in increasing the crop productivity itself. Moreover, it also introduces pest resistant, disease resistant, drought resistant variety to that of the plants itself. Okay. And like due to that of the recombinant DNA technology and all these things, the genetically modifications, by that of the genetically modification process or genetically modified things itself, automatically what is getting happened, these biotechnological principles, they always helps in producing that of many different kind of that of these kind of that of the plants, like the pest resistant one and all these things itself. Okay. Now, in that of the all the biotechnological products, we got one of the very special product, and that very special product we are representing it as of the single cell protein itself. That is the SCPs, the single cell protein, or that is the SCPs itself. Now, what is that? Actually, it has been observed that whatever the amount of protein the milk it produces, the, I would say, the double or triple amount of the protein can be produced by that of what, by that of cultivating the microbial organisms itself. Okay, when we are taking that of the microbial organisms, these microbial organisms, they are being cultivated, and when they are being cultivated, after they have been cultivated, they are doing what, they are releasing a lots of huge amount of that of the protein itself. They are releasing a huge amount of that of the protein, and all those huge amount of that of the protein itself, they are referred as that of the SCPs, or that of the single cell proteins itself. Okay. So if I'm saying now the single cell protein, now what is a single cell protein? The single cell protein, it is actually referring to that of the dead as well as another dry cells of the other microorganisms itself. Okay, such as we can say that the yeast, the fungus, the bacteria, and another algae itself. Okay. And if you're saying if this word, this single cell protein, this word, it was first of all been utilized by that of the person named as Carol Wilson. Okay, and that is also during that of the 1967 period itself. Okay, and we can say that this single cell protein, it is like fulfilling, currently it is fulfilling 10% of that of the world protein requirement, as I said you. Okay, like I would say that this cattle proteins, that means the milk proteins and all these things. Okay, they are how they have been produced, they have been produced by exploiting the animals itself. But single cell proteins, in which we are not exploiting the animals, we are exploiting the microbes which can multiply in a faster manner. So it is environmentally, I would say that it is environmentally better also. And it is like we can say that we can even show the preservation of that of the animals also by that process itself. Okay. Now, the climatic factor, it actually affects that of the agricultural process itself. Whereas, if I'm saying about that of the single cell protein, now these single cell protein technology, it will not be accepted by that of the climate itself. This will not majorly be accepted by that of the climate itself in that case. Okay. So if we are saying the advantages of the other single cell protein. What are the different advantages? The main advantage, the first of all, it is having high proteinous content. It is consisting of all the essential amino acids. For that reason, only protein content can be met. Along with that, it is highly rich within the other vitamin content also. Okay. The small doubling time, that means what? Like the multiplication time for the other microbes, it is very short. Okay. It can faster way, it can multiply and it can produce a large amount of the other single cell proteins also. Okay. Higher growth rate, it will be also having a huge amount of the high growth rate itself also. Okay. And that will be utilized in that of the flexibility in the use of that of the substrate also, it will be utilized over there. Now that will be energy efficient also. Okay. And cost effective also. Okay. And they can be mainly genetically controlled. These majorly they can be controlled genetically also. Okay. So now what are the different sources for that of the single cell protein? See, fungi, we can say in algae we can have spirulina as a major source. In fungi we can have fusarium, aspergillus, fusarium candida, penicillium, etc. And now here I had mentioned about that of the certain kind of that of the different other sources like microalgae. The protein content, it is 60 to 70, and we can say chlorella vulgaris, that we can say over here there. Yeast, if I'm taking 30 to 50, okay, production of vitamins and micronutrients, as well as use of variety of the feedstocks are remaining present. Saccharomyces cerevisiae, Candida utilis, okay, and then bacteria for that of the high proteinous content and growth on that of the typical kind of that of the substrate. That thing we are representing it as of the C1 substrate itself. There is a cuprovitus nectar, okay. Protist, if I'm saying 10 to 20% protein content, okay, as well as that a production of that of omega-3 fatty acids itself. And we can say that Schizochitrium limacinum, that will be remaining present over there. Okay. So we can say that like that of these are the major sources for that of the microorganisms also, and their like main selected criteria which are remaining present over there. So what are the different substrates that are suitable for that of the single cell production? Mainly the algae, the carbon dioxide, CO2, and sunlight itself. Fungi, it can be like sulfide waste. For fungi, there are the like waste, sulfide waste, then prawn shell waste, then dairy waste, whey proteins, molasses, lignocellulosic materials, and all these things, they will be the major part for major medium for that of the fungal culture. And if I'm saying in the bacterial culture, agricultural as well as animal and food processing wastage, methanol, and all these components. Now, the nutritional aspects of SCP. Algae, it is mainly rich in the other proteins itself, fats, vitamin A, B, C, D, minerals, and nucleic acids. So as I said you earlier only, when we are like, I would say that when we're extracting the protein from that of algae, automatically we have to know one thing, that when we are extracting the protein from that of the algae, it is always rich within that of the like proteins itself, as well as that of like the fat component along with that of the vitamin A component itself. That we have to remember. Okay. Now, if I'm saying about when we are extracting it from that of the fungi itself, in that case, so in terms of like that of the fungi, we will be having like B complex vitamins itself. Okay, along with that, the amino acid content that will be that will be very pretty much high itself. Okay, there are dry A's, it will be containing about that of what? It will be containing about that of the 50% proteins, as well as that of 20% of that of the non-proteinous components, it will be remaining present. The yeast, it will be containing that of the lysine, the thiamine, the biotin, the riboflavin, niacin, all these kind of the vitamins are remaining present. Now, if I'm saying about that of the bacteria, the crude protein content of that of the bacteria, it is around that of 80% itself of that of the dry weight. Okay. In general, the single cell products, it contains approximately 50 to 70% of that of the crude proteins. It is containing approximately 50 to 70% of that of the crude proteins itself, which is remaining present over there. That is for that of the bacteria in that case. Okay. Now, if I'm saying about the role of biotechnology in enzyme production, we have to remember that biotechnology, it involves the production of genetically modified organisms. Now, when they have been utilized in the genetically modified organisms itself, from those genetically modified organisms, the enzymes can be isolated or enzymes can be cultivated. Okay. It mainly involves the utilization of the microorganisms itself. The enzymes, it will be selected majorly as a result of the microbial activity. Okay. And in the form of the metabolites and in all these things, they will be secreted itself. Okay. And the use of enzymes, okay, that use of enzyme, it will be done at that of, I would say that the industrial level processing itself, the use of enzymes will be done. Along with that, the enzymes which will be produced, it will be consisting of that of the carbohydrates as well as that of the proteases itself. The enzyme that will be getting produced, it will be mainly produced as that of the carbohydrates along with that of the proteases itself, okay. And for production of that of the, I would say that these kind of the enzymes itself, okay, within that of a smaller amount of time, I would say that the cloning process and all these things, cloning of the gene, it has been done, or the genetical modification that has been done over that of this thing itself, okay. And now I am enlisting certain kind of the common genetically modified kind of the enzyme, the catalase, chymosin, new glucose oxidase, alpha amylase, protease. All these things are what they are the common, I would say that, like enzymes which are being produced. And all these common enzymes, they are mainly utilized in that of the mayonnaise productions itself. As along with that, if I'm saying about the chymosin itself, it coagulates out of the milk. Glucose oxidase, it is involved in that of the baking process itself. Alpha amylase, it converts out of the starch into that of the maltose, okay. And if I'm saying about protease itself, it will be helping in that of the meat tenderization process, baking, and dairy products also in that case, okay. So biotechnology and food safety, we have to remember one thing, that till now, okay, although the biotechnology had developed, the world had developed a lot, but till now there are lots of kind of things are present, okay, by which we are considering that of like biotechnological products as unsafe, okay. Like there are many in many where in many things that has been also observed that when the food product, it has been produced by any kind of that of, I would say, genetical modifications itself, due to because of that one, many of the individuals, they were even suffering from that of what, they were even suffering from that of, I would say, like the allergic kind of there are the symptoms and all these kind of there are the symptoms itself, okay. So because of which, like when the biotechnological part, okay, or any kind of biotechnological experimentations, genetically modified experimentation, genetic engineering processes are been done, they are been approved by that of a special kind of committee. And that committee, we are representing it as GEAC, Genetical Engineering Approval Committee, okay. So whatever the kind of experimentations are been done in that of the genetic engineering process or biotechnology processes itself, first of all, they passes on towards that of this genetically engineering approval committee itself. GEAC like approves these components, okay, or these products. And once GEAE approves these components or products itself, after which what used to get happened, after which I would say that this will be leading to that of the like, or this will be helping to maintain that of the food safety processes itself, okay. So now what will be getting happened, if I'm saying how this food safety will, like how biotechnology is affecting indirectly or indirectly, we can see that it will be like displacing or altering that of the genes itself, okay, we should be controlling the nutritional constituents, okay, of that of food crops, livestock, and all these things, okay. It can even alter that of the genes that will affect the levels of that of naturally occurring toxins also, okay. And by extending certain agricultural production practices such as the use of pesticides that lead to chemical residues in food and that of the water itself, okay, that has been done over there. That is for that of the biotechnology and for that of the food safety processes which are getting involved over there itself in that case, okay. Clear everybody? समझ में आया? I think so, yes. Okay, very fine. So we had discussed about today about that of the different kind of that of the fermented foods. I'm just like keeping a brief revision of the same itself. Okay, we had discussed about that of different kind of that of the fermented food itself, like idli, like sauerkraut, like pickles, like I would say that dosa and curd, yogurt, and all these productions we had already discussed. Okay, we had already said that whatever whenever these fermented products are being produced, these fermented products are always being produced in order to enhance or extend the shelf life of the individuals itself. So when the shelf life are being like extended or enhanced itself, so automatically we can say that automatically that will be leading to that of enhancement of that of the product quality also, and betterment of the product quality also. We had said about single cell proteins. The single cell proteins are what These are the special unicellular kind of the proteins itself, and those special proteins which are derived from the unicellular sources, it are mainly like rich within that of the proteins as well as the fats, vitamins, and all other content itself. Nutritionally they are highly rich, and they can be even produced in a very faster manner. Also they can be even produced itself, and for that reason nowadays the world, it is now shifting from that of the animal protein towards that of the single cell protein or microbial protein part itself in that case. Okay, we're also saying that like the genetical material, the biotechnology, it will be also utilized in terms of that of for enzyme production. The genetically modified organisms can be utilized for that of the enzyme productions also, and for all the other different things itself in that case. Okay, students. So I think so we are done for today. Okay, we had discussed all the components itself. We had discussed in pretty as much detail as it is possible. Okay, for further more details, for that of the further notes and everything, you can contact over the number given over there itself. Okay, best of luck to everybody. Best of luck for your preparation. Thank you, everyone. Thank you very much.